Yes, I think here we are again. Hi. Hello and welcome again. Yes, hi, hello and welcome again. Microbehunter here and welcome, yes, to another Saturday microscopy live stream. Happy that you joined in again. In case you're wondering why I'm always looking around at the beginning, is because have to sort out a few things here on my computer monitor. Yeah, until is running and until all of the housekeeping and all of the technologies is running here in the stream. Yeah, I'm really happy that you were able to join in again. Today I'm going to do again a lab together with you. I would like to put some pollen under the microscope. In case you're wondering what the setup here is I will explain. Yeah, I'm quickly going to check how many people have joined in. It's quite fascinating. Wow, 46 people. That's quite something. It's coming more and more every week. Very nice. Usually during the first couple of minutes, what I do is I simply go through the chat on the side to say hello all around the world. This gives the viewers a little bit of time at the beginning to join into the live stream without missing all too much. So there is hello from France. Yeah, please apologize if I happen to skip over one or the other one. Okay, from the United States, from Lebanon. From the Bodensee, yes, quite fairly close by. From Vietnam, Netherlands, Northern Germany, okay, Pennsylvania in the United States, Netherlands, okay, Oregon, United States. It's kind of funny because people are joining in even at the most impossible times. From the UK, Connecticut, London. Sound is good. Thank you very much. And I hope that the stream, it does not interrupt. A couple of weeks ago, the connection was pretty bad for whatever reason. I was not able to figure out why. From Norway, from England. Okay. From the United States again. I love tardigrades, so do I, but today we're not going to be looking at tardigrades. Philippines, from India, from Ireland, from Greece. I'm going to speed up a little bit. Yeah. Can you look at bacteria from your armpit? I'll be looking at bacteria. I don't know. I have to grow them first, and actually I do not like to grow bacteria from the body for safety reasons. But I have a few sources of safe bacteria as well. Okay. Will you show us how to control temperature when heating glycerin jelly? Yes, that's exactly what I'm doing right now. Okay. And how the slime mold's doing, and also hello from Sydney. So what I'll be doing right now is the following, very briefly. First of all, what you see here is basically the setup that you need to mount some pollen. I got the pollen over here. Yeah, I'll also explain from where I got that. But today I'd like to use the mounting medium glycerin jelly or glycerol gelatin. Not an easy to use mounting medium, but I'll take it slowly and I'll kind of share with you my experiences with it. And of course, maybe some of you have are way more experienced with that. So please feel free also to share your opinions. So that's basically what I would like to do. So we're going to take it slowly today, and maybe we're also going to do a little bit of slide ringing as well again. Okay. So yeah, I hope that I'm just reading every now and then the chat as well. If you have any questions, direct it to me, because sometimes there are quite a few hundred comments in the chat, which is great. But I would like to respond to some of them. So please indicate this by saying @Microbehunter or @Oliver, because Oliver, that's my first name. And then after approximately 1 hour, I will try to answer them. Okay. The on-topic questions I would like to answer right away. Off-topic questions we're going to delay until a little bit later. Okay, so I need to explain this here a little bit because you might be wondering why I've got this jar here upside down. Okay, and this, what you see over here, is a coffee warmer. This is not a USB one, but one that's actually connected over a regular power supply. You can adjust the temperature over here. This is a timer so that it will switch off automatically. And I put a jar over it simply to keep the temperature because it's going to be at 40 degrees. And I switched it on approximately 1 hour ago. To warm up and to liquefy the glycerin gelatin and the glycerol jelly. That's the mounting medium. So you see that I put 2 cups over it. So this is the one that I just recently ordered. It's been a request by one of my viewers to please show how to mount using glycerin jelly. And this is the one that I ordered. The company, I'm not sponsored, by the way. Okay, yeah, it's Brunel Microscopes. It's a British company, and they have pretty much everything that you want to have concerning microscopy equipment and chemicals and everything. So I've ordered from them already several times. And yeah, just a few days ago, I got the delivery again, including glycerin jelly, specifically for pollen. They've got 2 different types, the regular one and the one for pollen. I have to keep it on here a little bit to keep it warm. And it has to be molten. How do you see this? Yeah, you have to warm it up to 40 degrees and maybe maximum 55. But definitely not hotter. That's really important. If you overheat it, it's not going to turn solid anymore, right? And that's a problem, right? And this glycerin gel mounting medium is difficult to use. Okay, it's known to be difficult to use. First of all, because you need some kind of a warm plate, not hot, a warm plate, not too hot. If it's too hot, it's gonna break again. It's not gonna solidify anymore because the gelatin is heat temperature sensitive. It's extremely notorious to make bubbles. That's another problem. And last but not least, before I actually mount it, I also have to pre-warm the slide. Right? So you see that's still the easy part. I'm just going to give you an overview right now because I already made one of them today in the morning at my time. You can see it over here. So there is already some pollen over here. And now what I have to do is because it's a water-based medium and it does not completely dry out, I have to put a ring of nail polish around that. I didn't do that yet, but that's also something that I have to do. Otherwise, it's not going to be stable because this mounting medium does not turn 100% solid, but it's remains in a semi-solid state, and for this reason you have to put a ring around it as well. So you see that this is probably one of the more difficult to use mounting media. I'll just put this thing again over here to keep it warm, but it is the standard one that you use for mounting pollen, okay, and the results are quite nice. So what I will do every now and then is I will interrupt myself and I'll quickly have a look at the a little bit a look at the chat. So I'm going to just skip over. Yeah. If you want to communicate with each other over the chat, that's perfectly fine. But again, if you have a comment for me, please indicate that. Yeah. If there are some how do you say, issues that are directly on topic, I will try to answer them right away. One of them could be, doesn't Brexit make it difficult to order chemicals from the UK? In 2 ways, it's more difficult, and it's not only because of Brexit. I want to show this to you, so thank you for asking. First of all, I have to cough now, sorry. Okay. I'm better now. First of all, generally, it doesn't matter if it's from the UK or from wherever, liquids, like liquid stains, they don't send, right? So if you want to buy some stains, and look, that's also something that I bought, yeah, saffron in, you can buy it in powdered form, right? So that is possible. So that's a second dye, second stain that I've ordered, and they were not really expensive, honestly. Yeah, and the delivery was fairly prompt. However, because of Brexit, I had to pay some import tax, but that's fine. Yeah? So that's a little bit the thing. But because these are powdered form, it was not a problem. But indeed, they will not send liquids. But that's not only an issue because of Brexit, but it's generally an issue even on Amazon. They don't send liquids, right? So I just wanted to show this to you that I've basically also bought a couple of other stains that I would like to experiment with. But the glycerin gelatin, because it was kind of solid in gel form, it was also not a problem. Okay. So then other questions over here that I will answer later. Okay. So I'm quickly just going to scan through here. Yeah. Yeah, I'm just going to skip over and I'll only answer the on-topic questions. Yeah. So another question, you heat up the whole package of glycerin jelly. How often can you do that? I read somewhere that it's better only to heat up a small amount. Yes, that's correct. So I will quickly tell you why I decided to heat up the whole thing What you can do as well is you basically take a little bit something like a spatula or so, and you scoop out a small amount from the solid glycerin jelly and you melt it directly on the slide. This is actually also something that can be done. I'm not going to do that because I just want to always transfer a consistent amount of 25 microliters. So that's simply for practical purposes that I would like to do it like that. Okay. But you know what I'm going to do, because I do have to prepare the pollen, and the pollen has to dry on the slide first, and this takes time. So what I think is, I'm not going to talk now much more. I'm just going to get started. Otherwise, there is going to be too much waiting time. So the pollen basically is here. So, and I need to explain what this is. I'm going to lower the camera again, because what you see over here, thank you again. I don't know if you're watching, but I got this a few months ago from a friend in Germany, from a microscopy friend in Germany, who's basically, from a bee, they've been doing some beekeeping. And these are the so-called the pollen baskets. These are basically packages of pollen that the bees carry on their legs. And I got those. And so basically that's concentrated pollen, really great. In springtime, right now it's February, so Central Europe where I am, it's still winter. But in a couple of months, it's going to be springtime, and then I'll be collecting some fresh pollen as well. So that's number one. And number two, I've got those little Eppendorf tubes that prove to be very practical, and I'll be dissolving a pollen basket in some water in here, and then I'll be using that to make the slides. Okay, so I don't know. Let's go up again here a little bit. Let's turn this up. Let's say I'm going to use 200 microliters. I think let's use 200 microliters this is distilled water, but it's 1, 2, 3. So 200 microliters should be more than enough, actually. Yeah. And next thing, you always move the water away so that it does not fall over. Now I'm going to take one of those pollen baskets, and maybe you see they have different color even. And I'm going to just take. I'm going to pick a random one. Yeah. And I'm going to just pick this one. That's a lot, actually. That's a huge one. And I'm going to put it in here. Here it is. It goes in. Oh, here's on the side now. And I'm going to dissolve it. And I'm going to take some of this liquid then and put it on a microscope slide. And in order to dissolve it, let's move it up again a little bit here. In order to dissolve it, I simply push it into the water. And I simply draw it up and down. This one actually dissolved quite quickly. No, it's still here. The one in the morning, the one that I had in the morning really was not so easy. But here's still some clusters. Okay. Actually, it's interesting. I can actually see it much better on the monitor than than in real life. Let me refocus it. My camera has this out of focus button. Yeah. And maybe I'm just going to do the following. Yeah. So that's going to be a lot of pollen. And there is a book that I also bought, which I also want to show you, which recommends that I do not suspend it in water. I mean, suspending it in water is one possibility, but it also says you should actually suspend it in 50% glycerol, because the mounting medium also contains approximately 50% glycerol. However, there was a problem with that when I tried that, because the glycerol would obviously not dry out completely, because it likes to attract water. So I think you see now that this is seems to be okay. I mean, there's still some clusters in here. I think it's fine. And what I found out is, you know what, I just wait for a minute or so until everything's properly suspended. And what I'm going to do then is I'm going to put 25 milliliters on those slides here. And then we have to wait until it dries. And I just want to show you now the other slide, the one that where I try to prepare pollen with 25% glycerol. You see that it does not dry completely, obviously not, because glycerol likes to attract water. right? Yeah, so, but I would like to try it basically with a completely dry slide, so I'm just going to use pure water, okay? So, yeah, I'm going to wait a minute or so for this whole thing to completely suspend, and I'm just going to occasionally read some of the questions here. So why are we using glycerol specifically? Yep. So this one here is glycerol gelatin, contains gelatin and water and glycerol, and a dye, the red dye, and some disinfectant that prevents it from becoming moldy. The reason why glycerol is added is because it should not completely dry out. Normally, mounting media should completely, the solvent should completely evaporate and dry out, and then it's stable for maybe even 100 years. But when you make a slide with glycerin jelly, it does not and it should not completely dry out. Okay, and the reason is because if it were to dry out, then the pollen would shrink. And you don't want that. You want to have the pollen in their kind of natural state, and then there must be water present. So the glycerol jelly is somewhat of a way of permanently mounting slides without the mounting medium completely drying up. Okay, so glycerin gelatin is also used for mounting delicate water organisms, for example, like paramecia and delicate algae and so on, because if they were to completely dry out, then they're going to shrink and they're going to lose their shape. So there must be water present, but at the same time, it should be kind of solid and stable, and therefore they're using glycerin gelatin, and the glycerol prevents it from completely drying out because it attracts water. Right? But you see, if it's not sealed off here on the side, then this basically means that moisture can enter and exit as well, and also spores from fungi, and then basically it's not stable. And because it's not completely hard, and also the cover glass is not completely stable, so you need to put a ring around it, and that's what I'm going to try to do later as well. Yeah? So that's why it's a little bit a special purpose mounting medium, and they have got different recipes specifically for pollen and for other things. Yeah. So the one that I've got here is specifically for pollen. Yeah. So I'm just going to read this, and I'm just going to skip over all of those questions that are not directly relevant, and we'll try to answer them later on. Okay. Yep. There are lots of comments here. That's fine. That's great, and I think. Discovering the pollen bundle wireless could use an old ultrasonic toothbrush to agitate the sample quickly to basic, don't know, not ultrasonic. Well, toothbrush maybe. Okay, you got to be careful. You can agitate this using a toothbrush, certainly, but if it is real ultrasound, then essentially you've got to be careful, because real ultrasound is actually used to break up the cells themselves. Okay, the real ultrasound has the same resonance frequency apparently as the cells, and is actually used to actually break open cells. So just be careful that you're not actually destroying the pollen, right? But I think a normal toothbrush is still way slower than actual real ultrasound. So I think it's going to be enough now. What I'm going to do now is the following, and this is also one of the things that was recommended, and I'm going to prepare a few, maybe 2 or 3 slides here. I have to see if I'm able to, yeah. And what they have said is that you should actually wipe it off with alcohol to make sure it's free of fat and oil and grease. So I've got a little bit of, so this, you can see this is some ethanol, yeah. It's basically just a plastic bottle with some ethanol here. And I'm going to simply not dry wipe it now, clean it. Of course, the problem when using tissue paper is that there are always these fibers, the cellulose fibers as well. So that's also another issue. And the last one, because apparently if it is not completely clean, then there is the danger that it does not stick properly. I mean, it's generally always a problem, but maybe especially with the glycerol jelly, I don't know. So, and now I'm going to just put this over here. I'm going to now use 25 microliters. Why 25 microliters? Because this is what I simply decided to work. I tried it out today in the morning and it worked. So, yeah, And yeah, let's try that. Yeah, so let's take 25 microliters. Let's make a little drop here in the center. And you might, yeah, the pollen themselves, they're not always the same amount of pollen. It's a little bit out of focus again, because the pollen being more dense than the water, they also settle a little bit down to the bottom of the tube. So, and now I have to wait a little bit until it kind of dries. What I'm going to do, how am I going to speed this up? I don't know how long it's going to take. Otherwise, I'm going to simply remove the glycerol gelatin and maybe go up with the heat a little bit. But let's wait for 5 minutes. If it doesn't work, then essentially I'll go up with the temperature. But I have to remove this the glycerol jelly first, because, as I mentioned before, if it becomes too hot, it might not solidify anymore. Okay, so I'm going to use this time again to go a little bit through this one over here. Instead of dissolving with distilled water, you can also use alcohol. It's often used with the pollen itself. Distilled water is often used when extracting pollen from honey. Yes, and as a matter of fact, the alcohol is also recommended in this book here. Because the alcohol also sometimes takes away some of the pigments, some of the stains, not stains, but some of the pigments that the pollen have. I think it depends a little bit on the pollen, because if you use maybe too much alcohol, maybe it also starts to shrink. Yeah, so I would say it's probably a question of trial and error. I'm not going to show you the contents of this book here, okay? But basically, I also ordered this from Brunel Microscopes. It's basically a booklet rather, a thin book that essentially, yeah, does exactly that, what the title is. It simply gives you advice and some protocols and how to collect pollen. You might wonder a little bit, who would even collect pollen? Why would you do that? Is there any meaningful... Yeah, of course, people who, for example, are beekeepers. You can, by extracting the pollen from honey, you can know which type of plants the bees have visited. It's used in forensics. Yeah, of course, we hobbyists, we like to do that as well. So that you see there are a variety of reasons why pollen actually are investigated, and why there are quite a few resources available on pollen microscopy. Yeah, especially for plant identification. But what I heard is it's not so easy to identify plants over pollen. Okay, it's not the easiest type of activity. Yeah. So whenever I use paper towels, yeah, that's correct. You know what? You use a microfiber cloth. If you have those fibers on the paper towel, use these are microfiber cloths used for cleaning eyeglasses. Yeah, and this actually works quite nicely. Okay. It would be nice to have more observation microscopy live streams looking at water samples. You know what? Yes, I actually originally planned to do this as well. I can show you. I've got actually several water samples that are collected from a pond the other day. Not today, I decided, because I just wanted to try out the pollen as well. Yeah, I'll try this as well. But you see, the water sample is always a little bit of a an uncertain thing, because sometimes you don't find interesting things. Yeah. So it's slowly starting to dry. However, what I'm going to do now is I'm going to speed this process up a little bit. I'm going to put this now to the side, and maybe I'm going to go up with the temperature here. Okay, or I'm going to remove this rubber surface here to increase, and maybe I'm going, so 40 again, yeah, and so. A few other things. If you have been watching my this channel here, you've seen that over the last couple of days I uploaded every day a small video. I'm experimenting around with different formats, unedited, almost unedited videos. You know, leave your comments on how you like this format. I'm experimenting around, right? I've basically now tried to let's put this with slightly more experimental videos, unedited videos, on my channel. Basically a little bit in the format as you're seeing right now. Yeah, I wonder how you like those, and would be interested in a little bit of your feedback. Sometimes I included music, sometimes I did not include music. I don't really know what works all the time, or what the viewers prefer. Usually they're unscripted. I was also thinking about making some scripted videos. Yeah. So yeah, give me a little bit of feedback of what you would like to see. So I'm always very thankful. Yeah. So the study of pollen is also used to reconstruct ancient environments and document environmental changes that had significant impacts on past human societies. Yes, I also heard, for example, if you by studying also fossilized pollen, they know which type of plants lived there and a progression of plants. So changes in the fauna and in the flora rather in plants and so on. Yeah, archaeologists use pollen from excavations to reconstruct the landscape from the past. Yes. Yeah. Okay. So, have you ever tried to mount pollen with PVA Elmers, or would this be nonsense? I think I once tried it. Of course, it works. No, it's not nonsense. The idea is simply that it's simply a non-standard medium. I would say almost everyone who is doing for pollen microscopy, they're using glycerol jelly. And I think the reason is because it simply maintains the shape of the pollen best. Yeah? But it also with a refractive index, it seems to work quite well. Yeah, see, it's already starting to dry. Okay, so it's quite nice that this is starting to, yeah. I have to make sure that this one is also still liquid. It's still liquid, that's good. Okay, I have to be a little bit careful about this as well. Yeah, so maybe I'm going to put this here, also on the side. Yeah. So, and it was actually as I mentioned, you really have to be careful. You're not allowed to move the glycerol jelly too much because it immediately forms bubbles. And even in the book, it says here that once you've got bubbles, they're almost impossible to remove. Yeah. I found it to be okay, but one has to be a little bit careful. Yeah. Do we need to do a similar procedure if we collect the pollen directly from the flower? And why are you not doing the same? What advantage will you get from pollen sac other than study variety? Well, the simple answer is this is one surprising answer. The reason why I'm studying the pollen, those here, is because it's winter time. Where I am, so I don't have access to fresh plant pollen right now. Yeah, and these ones here, they really have a high concentration. So this is for demonstration purposes. This should actually work quite well. Yeah. So I actually see that this one over here is actually already finished. Yeah. So this one actually is quite fine. I put it over here. This one over here, I think as well. I mean, if there's a little bit of water, I guess it's gonna be fine. Yeah. And this here is as well. Yeah. So maybe it's not a good idea if it completely dries out anyway. The reason being that otherwise, maybe if it's a little bit moist, then there's probably gonna be less air. Fewer air bubbles. Yeah. So over here, I'll keep it on here again, and now this is where the fun starts, because this is now where we have to be a little bit patient. And look what I've got. Where is it? I've got some round cover glasses, and I already cleaned those also with alcohol. Yeah, probably shouldn't touch them, right? So it's pre-warmed now again, and actually I should also pre-warm those. That's also what one has to do. And let's give it a try now. Okay, I'm going to take a fresh tip from the pipette. So, and let's give it a try. Yeah. You can already see there are quite a few bubbles. Let's make it bigger. Here we go. Now the camera is a little bit in the way, so let's see if we can. Yeah, and there is already a stain included. Yeah. You see, this bubble over here is not going to be, I have to take one of these here carefully from the side. And I hope I only have one. And carefully lower it on here. Usually the bubble is going to burst, I hope. And is it gonna burst? Boom, it burst. Very nice. Okay. And then you can actually see that the refractive index is such that you can almost not see the cover glass anymore, right? Yeah. And basically you have to let it cool, and then you can put a ring around it. Okay. And after a couple of hours or so, you're actually going to see that the pollen grain, they will turn red because of the stain. Like for example, this one over here, which I made a few hours ago. We can compare it then later. Yeah. And this one I'm just going to put now to the side. And I'm going to make the other one. Yeah. So I'm going to put it over here to the side. Next one over here. I do hope that the tip still is able to... Now, see? It already turned solid. It's not able to take up anything anymore, right? So I have to exchange the tip. So... let's give it another try. Yes, here we go. Yeah. And another cover glass. Let's try it. And let's hope that this works. Yeah. Yep, the bubble burst, and again we have. Yeah. So the second one, and let's try the third one as well. Yeah, let's give it a couple of seconds to warm up again. Let's see if the tip still works. Nah, it doesn't work. Yeah. So, I always have to exchange it. So, and the last one. Let's yeah, there are more bubbles here. Let's see how this works. The bubbles seem to fuse together now, and maybe it's also going to be pressed to the side. Let's see. Up, yes. And let capillary action do the rest. Okay. Yeah. So I'm going to just close this here again. Okay. And what we can now try to do is we could actually try to put it directly under the microscope before we start ringing it. Okay. I'm going to quickly check again if there is some comments over here. I'm kind of a little bit worried that I skip over something here. I guess the pollen has a tube cell. Okay, I'm going to talk about those. No, you cannot see them under the microscope because the two sperm cells that are formed actually form inside the pollen tube. And there is another comment over here, which I'm just going to respond directly. I'm a retired cook and do a lot of brewing. I have leaf gelatin and glycerine. What dye would I need to make the mounting media? I also experimented with leaf gelatin. However, what I heard is that, I mean, you can try it experimentally, but apparently the pH of the cooking gelatin is not always correct. Yeah, so you have to be careful. It's not exactly not laboratory grade. So you do not have to add a dye. That's not necessary. There are certain recipes. If you Google glycerin gelatin online, you're going to find several recipes. And the thing is you do not need to add a dye. Okay. This one just has one. You can see it perfectly without dye. But what you should add is some kind of a disinfectant. And I've read somewhere online that some people, when they made this, they added basically a mouthwash, Listerine. It's the mouth rinse wash, right? A few drops to prevent this from turning moldy and bad. Yeah? So there are some experimental protocols out there as well. Generally, there are two different types of common glycerol gelatin formulations. One is called according to Kaiser, and one is according to Kisser. And what you have to do, the one for the pollen is usually the formulation according to Kisser. And the Kaiser is the regular one that's used for water organisms and so on. Huh? So, but you know what? Let's do, as requested, a little bit of observation as well. So this means I have to switch over and I have to find the microscope view. Let's turn off the arrow. Let's turn on the light here. So, and I'm going to put one of these now under the microscope. And let's hope that we're going to be a little bit successful. I mean, we have already got a few pollen here. And then I'm going to compare this with the one that I made before. I mean, the density is not really high. I have to tell you. It's rather a little bit, yeah. You know what? Let me try the other one. I was actually hoping to find a higher pollen density. Here are some more. Yeah. Here. That's something like this, maybe, right? Yeah. Yeah. So that's basically now the 20 times. I need to turn up the light. And this basically now tells you what I meant with bubbles. Because those round circles that you see here, these are of course all bubbles, right? So, yeah, but if there are not too many of them, then it's going to be fine. I don't know what the things here in the back is, if there's some kind of a contaminant. But the thing is the following. This is now regular bright field. I'm going to now switch on DIC. Of course, it looks much nicer like this. Let's add a little bit of color. Okay, just for the fun of it. Okay, yeah. And the refractive index is just right to make the pollen visible, and you can see that the pollen is nice and round, because if pollen that's too dry, it will start to shrink and shrivel up. I've also once tried to mount pollen in Uprol. Some of you might remember that Uprol is also one of my favorite mounting media. But it contains a solvent, and this not only changed the shape of the pollen grain significantly, but also caused it actually to bleed out some of the color, some pigments that the pollen had. So just for your, let me quickly do the following, just for comparison. I'm going to remove now again the polarization, and I would like to show you how it's going to look like after a few hours, because I'm going to remove this now carefully again. Because the pollen will start to absorb the pigment, the dye. Okay, I need to quickly remove this here. So this is the one that I made some today in the and exactly the same procedure. But you notice that the pollen now, looks more reddish, pinkish, right? And that is because it's had now a sufficient time to absorb the red dye that was added to the glycerin gelatin, and we could not see this yet with the freshly made slides. You can also see quite nicely that it's more red on the outside than on the inside. Yeah. So it's actually something that I could watch. I could actually watch the pollen become more and more red. Yeah. Over time. Yeah. But again, this is not. It's not necessary to have this. The pigment in there. Yeah. So quickly, as we watch here, oh my gosh, there's so many comments, really cool. But I'm a little bit worried that, I'm a little bit worried that I'm overlooking some of the questions. I hope that, I'm going to get another list with questions later on. Yeah. Why did the glycerol in the tip become solid? I thought glycerol was not usually, it's glycerol gelatin. It's the gelatin that makes it solid. It's a mix of water, glycerol, and the gelatin. Yes, there are also some mounting, like for example, you can also mount certain specimens in pure glycerol. Not all of them are suitable to be mounted in pure glycerol, but then that would remain liquid. But what I want actually is I want the slide here, right, to actually to the cover glass to actually become semi-solid, right, and so to stick to the glass slide, and for that you need the gelatin. Yeah? So that's the thing, and the glycerol prevents it from completely drying out. Yeah? So it should contain some water. So this glycerol gelatin is kind of actually is a mounting medium that becomes semi-solid. Yeah. So let me see. I'm just skipping ahead. Maybe you can warm the tip of the pipette. Yes, I could do that. But yeah, for simplicity, I'm not. Yeah, the problem with the pollen and diatoms is that they get carried around by wind for long distances and stay airborne for a long time. So in archaeology, it's not always a good biomarker. Ah, okay. Yeah. So some comments here. If you mount the pollen with PVA and then wring it before it is dry, will it stay liquid? Yeah, I would say probably yes. I've never tried that. Okay. Yeah. There's a question on Canada balsam, which I'll also try to answer later on. Yeah. So that's basically what I wanted to show you. That's basically how a slide looks like. That's already a couple of hours old. And again, let's compare it with the one that I just made now. Yeah. You can see that this one still is not quite as pink. Yeah? And one of the nice things here is that the pollen, because the water dried first, the pollen is completely flat, and so kind of sandwiched a little bit between cover glass and microscope slide. Yeah, so that's Let's go up a little bit with the magnification again. So I'm now in bright field, just for your info. I've never tried it. This is now 40 times bright field. I'm going to try phase contrast. I've got a 40 times phase contrast. Let's try that. It's first time ever that I'm trying it. The pollen. Yeah, also nice. So that's now I've got a 40 times phase contrast objective as well. Yeah. It's also okay. Yeah. Yeah. So that's also quite nice, huh? So, by the way, I want to do a little bit of another thing that I want to just share with you a couple of other ideas that I'm having. I mean, one of the things that, as I mentioned before already, is that I've made those, quick videos now every day for the last week. I want to try to continue and see how this works. But I was actually also thinking of actually doing some just Q&A sessions to kind of summarize all of the questions that you were asking during the live streams, maybe to simply collect them together and make separate videos of that. So it's a little bit more also like a podcast format. I'm thinking about a lot of things. Some of you requested a few weeks ago also some regular biology lessons. I'm still thinking about this. I've not completely discarded the idea. I mean, I'm doing quite a few online biology lessons for my students already in school. But these lessons would not quite well fit with the microscopy channel that we've got here. So this is still something that I'm a little bit considering how to best do that. So let me quickly read. Every time when I'm pausing, I'm just reading some of the comments that you've got. Yeah, so this is kind of a phase contrast, and yeah, that's dark field. Yeah, I've got dark field only with the 4 times objective. It's also quite nice. Yeah, so I'm basically using now the phase contrast annulus as a dark field. Yeah. As a dark field ring. Yeah, so and that's now again in bright field, and and that's the one again that's a few hours old that I made today in the afternoon, my time. Yeah, and you see that they are much more red because they have absorbed more of this pigment. Yeah, in the book that I've in the screen book, yeah. It mentions that it's possible to also to stain it first and then just mount it in glycerin gelatin without a pigment. So there are a variety of different possibilities, or you don't have to stain them at all. Sometimes it's a good idea to wash them in alcohol. Some people stain them and then wash them. So I think there is a whole range of different variations to the whole protocol. Yeah. So what I'm going to do now is something that scares me a little bit sometimes because it's a little bit messy, and that is, I can already switch off the coffee warmer. It's a little bit messy, so I'm going to take this away. Yeah. Yeah, And let's put it away. The thing is the whole thing about slide ringing. Okay, let me put the coffee warmer away because I do need the space. So let's put it here. And yeah, it's not the first time that I'm doing this in the live stream. Maybe some of you have not seen it yet. Let me trash the tip. And that's the following here. That's my slide ringing table. Yeah, some people have actually made advice you don't need to buy one. You can actually, there are a whole range of homemade do-it-yourself alternatives using CDs that are spinning on a fidget spinner. People have 3D printed those in the past. Some of them actually motorized them. Yeah, there are so many variations and pictures that also some of you have sent me in the past. So yeah, but I in any case decided to buy this. I also got this from Brunel Microscopes. Again, I'm not sponsored by them or anything. I just think it's cool that they have these things, right? And I'm just going to now do the following. I'm going to try to make a ring. And this is now, I made this black ring here using a felt tip pen. And normally you're supposed to use a brush, but I'm not. I'm going to use the pipette. And this is usually the highly, how do you say, delicate issue. Because, I tell you what the problem is that I have. I don't even know if it's properly centered. That's a little bit the problem, right? Because I cannot see the how do you say the edge of the cover glass anymore. That's a little bit of an issue because the refractive index of the glycerol jelly and the slide are so perfect that I don't see this anymore. Yeah? I think it's I don't know. Yeah? That's actually something that I did not entirely expect, that it's that difficult to see. But I think this must be now kind of centered. I'm going to look. I'm going to try it in any case. If it doesn't work, then well, so be it. Or I could try to kind of touch. See, it kind of goes out here. I have to move it in a little bit further. So I'm kind of slightly touching the side here, almost like a record player here. So I have to move this in a little bit here. Yeah, this is now reasonably centered. Okay. Yeah, so I'm kind of scratching the edge of the cover glass using the tip of the tweezers. So normally what you're supposed to do, you're supposed to take a little brush I'm using always, I always like to use nail polish, okay? But what I found is I found that it's actually sometimes easier to simply use a pipette. Okay, it allowed me, at least in the past, to deposit a more controlled ring of nail polish. Yeah? So, and we're just going to give that a try. And usually this is can be a little bit messy and really takes a lot of patience. And I'm doing this now on video and live and without my glasses. And so this is maybe more a proof of concept than anything else. Okay. And I have to go all the way in here. So here we go. So, and I'm going to now spin the whole thing here. I'm going to give it a little bit of and I'm slowly pressing it out. Yeah. And that's how it looks like, and I think it's not enough, okay? So I'm going to try this again. So maybe a larger pipette wouldn't be bad. Normally you would do this with a brush, but I like the pipette method more. Yeah, it's slowly already starting to dry a little bit, and for this reason it's not flowing quite as expected. But I'm going to discard this here again. But maybe, let's have a closer look. it's not flowing very nicely. Okay, so maybe I should add a little bit of acetone, but I think you get the idea. I do have a second nail polish here, black one, so I could try this as well. And the point of this ring is, and that's actually necessary when you're using glycerin gelatin, is not only to stick the cover glass to the slide, but also to kind of seal it off so that no water moisture is able to enter and exit from beneath the cover glass. Yeah, no, honestly, I've made already nicer rings before. This looks a little bit very scratchy. It should be a very smooth, round ring. Okay, yeah. Okay, there's an interesting. Marcel just commented over here. Sealing with nail polish will extend the quality of the slide with a couple of years. Normally, without using nail polish, you can keep the pollen slides about 5 years. Okay, yeah. Yeah, so I think it depends maybe also a little bit on how the slides are stored, if there's a large variation in moisture or not, maybe also on the amount of glycerin jelly that was used. Yeah, so but I'm not entirely happy with this one over here. I just realized, so maybe I should buy myself some more nail polish. Yeah. So yeah, it's too. I mean, it does seal it off here on the side. Yeah, I mean, that's important, but honestly, it's also not a lot. Yeah, so let me try this again, yeah, with the next one. So here and here, I have to kind of center it again. So let's see if this is, yeah, here it's too far out. Yeah, it's kind of a little bit, yeah. So let's give it another try. I hope that the next one actually will. Maybe I've got here, that's the black nail polish. Okay. Here. So I don't need the brush. There seems to be a little bit more in here. It's going in all the way. Yes, let's try that. Ah, nothing comes out. That's a demonstration effect. That's a demonstration effect. Okay, that's a little bit of a pity. But I do have another idea. Maybe yeah, I should add a little bit more acetone. But sometimes we learn a lot when we are not successful. I'm going to try now the following. I'm going to simply make it a little bit larger here, the opening. And let's try this again here. So, and it's a bit better. It's a bit better. So basically, by cutting off the tip of the pipette, that's too much. Okay. Yeah, but that's definitely a very solid seal that we have here. Okay, it's a little bit I just want to show you a little bit how this works, right? And then you try this out on your own. The thing is I tried it in the past a lot with using a brush, and it did not work quite as well, I have to tell you. I'm actually really, even if this ring is a little bit thicker than normal, I actually did get more reproducible results this way, and or you apply a thin ring first and then allow it to dry and then apply another one. But yeah, so just showing you how it works. Let it dry, because if you accidentally touch the microscope objective with this, it's not the best thing. So yeah, where is my? I still have some of these slides here. Let's have a look at this slide here. The one that I just made as well. Where is the scope view? Let's see if it's already slowly starting to turn red. Yes, and I hear that slowly the questions are coming. I cannot believe this. Really? there are more and more questions. Wow, 3 pages, almost 3 pages. Thank you. This actually is a sign that there are more and more people joining in into the stream. It's nice. I'll try to, yeah. And you can see these are basically the slides which I just made. And what do you notice? What do you notice? You notice that they're slowly starting to turn red from the sides. Yeah. So yeah. Basically the pigment, the stain which is here diffuses and binds against the pollen, and that's why the pollen starts to turn red, yeah, from a little bit from the outside inwards. So I'm just occasionally looking over again to the could you rim the edge of the cover slip with a black marker to help with visualization? Could you rim the edge of the cover slip with a black marker? That is a really nice idea. Thank you for you see, that's also one of the reasons why I like these live streams, because I myself am learning so much. And you know what? We're just going to try out. Honestly, I'm just going to try this out. This is a really nice suggestion. Where is the desk? I don't know if I'm going... This is already turning solid a little bit. Yeah, it's slowly turning solid already. You know what I'm gonna? I don't know. Yeah, I'll try it in any case. I don't have now. I'm going. Look, I've got this slide where there are some pollen on here using glycerin. Okay, and I'm just going to recycle this slide here, and I'm going to now do the following. I'm going to. But it's not warm anymore. I have to warm up the coffee heater again, otherwise it's not going to work well. So let me see. Yeah, I have to be a little bit careful about this. Let's try it. Let's do number 1, this here. Number 2, where is a? Do I have a marker? Let's try that. I don't know. This is a. I know I'm not supposed to touch it because of the. Yeah. But maybe, I don't know if you're able to see that. It's not here on the edge, not. But maybe if I do it like this. Do I see something? You can, yes, you can see it here a little bit. I don't know if this is enough or not. I mean, this is a blue marker, right? So it's not going to... it's a blue marker, so it's not going to, the contrast is not quite as high. You can see it here on I dropped it. Okay. Yeah. But that is essentially indeed a possibility maybe. Yeah, I'm just going to quickly leave it as it is here, and let's see if we can do that. Yeah, it's not turned too much. That's very little. It's too little. You can actually see the ring here. Okay. So actually labeling it with a marker should be possible, but then I just show you. Let's put this away. Let's put it away here. But then I should probably remove the ring from the slide ringing table. Yes, it would work. Okay. Yeah, if I were to use a proper black marker, make a proper ring here, and remove the black ring over here with some alcohol, then actually, yeah, thank you for the recommendation. I think that's indeed a feasible possibility. Okay. So what I'm going to do now is the following. I'm going to dive into some of your questions. Okay. So, and as always, this is not going to be. I mean, it was the first time that I'm experimenting with glycerin gelatin. I promise you, it's not going to be the last time. I'll of course be making videos as well. Yeah, and then basically. maybe I can report back when I'm a little bit more experienced with these things here. So what I'll do is I'll quickly, because look, this is really nice. Never before did we have so many questions, like 3 pages. I'm going to quickly answer some of them. Some of them I might have already answered. Okay, then I'm going to skip over them. And maybe some of the answers I might not know. Okay, and then I'm going to quickly go again into the chat. Maybe there are some remaining questions, and I think then it's already 1.5 hours anyway. Yeah. Will you show us how to control temperature when heating glycerin gelatin? Yes, that is essentially this one over here. I have to, however, tell you that I think that this becomes hotter than what the temperature display is. So I do not entirely trust the temperature display. So I suggest that you do the following, that you put a cup of water on here and then measure the temperature of the water. I'm saying this because if it becomes too hot, then the glycerin gelatin is going to break. It's not going to turn solid anymore. So you have to be careful. So don't trust the display. How are the slime molds doing? Yes. How are they doing? I just made a video today as well. Here it is. Okay. This is a little bit... I got some cookies from my students. Chocolate cookie and a regular cookie. And this one is some oat cereal. And you can see that the slime mold prefers the cereal. I was thinking that maybe the high sugar concentration, somebody also suggested maybe high fat concentration, oil concentration of the cookies might have maybe the slime mold doesn't like those. But look here, the slime mold goes to the cereal. Yeah, so slime molds are doing fine. Also something that I would like to show you here. Where is it? Here, that's another one. On filter paper, you see how it's dried up. Also here, it's called the sclerotium. And basically, I'm going to cut it into small pieces and for storage. And then I'm able to add a little bit of water again, and it's going to come back to life. So this is basically something that I wanted. It's because they're able to survive dryness as well. So that's a little bit the slime mold thing, but I did make a few videos, so please have a look. And I just got a micropipette today, although the tips won't stay on. Any suggestions? They should stay on. Honestly, then maybe you got the wrong size of tips. Yeah, but they should actually stay on like this. And then when you push the button here, which you cannot see because it's too, and if you push the button here, then pew, it should fall off. There's some, I have to clean this because there's some blue nail polish on here now. So, that's the Brexit question. I have to pay import tax now. That's but that's a little bit an issue here. Are Brunel microscopes any good? There's so little info about them on their website. I mean, what's a good microscope? That is a philosophical question. I mean, honestly, I never bought microscopes from Brunel. Only other equipment. But they have got people that you can talk to, right? And if there are issues, I'm quite sure that they're going to be willing to help you out. I mean, that's actually one of the good things if you actually have under quotation mark, a real shop. And because you can actually have people there that actually you can contact and you can talk to, right? And and whether a microscope is good or not, it does not only depend on the microscope, but to a large part also on your own expectations. I generally recommend that when you want to go into microscopy as a hobby at the beginning, maybe not to spend too much money and then simply upgrade later on. So that's by the way, I'm going to be making in the next couple of weeks or so. I'm going to be making again review videos about microscopes from the company Euromex. It's a Dutch company. And they've got also some very nice devices. I was wondering if you may be aware of this fantastic German microscope designer. He designed compound microscopes with glass lenses with 250 times magnification. Honestly, really? Really, 250,000 times word, that's electron microscopy. But then again, if you want, you can have any microscope magnification that you want. It's just going to be more blurry. So essentially, 1,000 times is the maximum. So anything above that is called empty magnification. Yeah, there's a physical limit to that. I cannot remember the man's names or his company's name, but he was a German. However, he passed away several years back. I mean, there is this, was it Rife microscope? He claimed to have magnifications up to 100,000 or so. But that's actually not correct. Yeah. I know that there was this one, I don't know if it was German or not, but I know that this one, he claimed that he can see viruses with a microscope, optical microscope, but it's not, unfortunately, it's not correct. You heat up the whole package of glycerin gel, yeah. How often can you do that? I don't know. I only know that essentially the glycerol, the gel will degrade, the gelatin will degrade if you overheat it. Yeah. So yes, heat up small amounts. I agree. I really have to agree, and I've seen some protocols that actually say this: you should take out a small piece of the solid gel, cut out a small piece, and then heat it directly on the microscope slide. Yeah. Glycerol is used in the jellies to prevent complete evaporation of the water because it has to stay watery. Dissolving the pollen bundle while a small centrifuge tube. You can use whatever you want to shake for shaking. I think a toothbrush itself is not going to vibrate with ultrasound as such. Yeah, if it's really at the so-called at the resonance frequency of the cells, and the cells are going to break open. But generally, I see no problem. To use this otherwise. Yeah. How long have microscopists used this medium? I don't know, but I know it's one of those traditional media, and it's been around for a fairly long time. I think it might have been even one of the earlier mediums ever used. Yeah? But I don't know so much about who invented this, but I know it's been around apparently for quite a long time because glycerin has been around for time. Gelatin has been around for quite some time. Yeah. Have you ever tried to mount pollen with PVA? No, I wouldn't say it's nonsense. I've tried it, but I don't remember how good the results were, right? So I see no reason why it should not work, but because PVA completely dries out, for this reason, I can imagine that maybe also the pollen grains will start to shrink and change their shape. Yeah? Can you use the glycerol in the cover slip and seal it with something to make it oil and water resistant? Can you use the glycerol and cover slip and seal it with? I assume that you're trying to you make a slide using pure glycerol without any gel. That also works. However, and I've also read something about that. However, that is difficult because you have to essentially seal it on the side. But the medium that you use for sealing should not touch the glycerol. Yeah. So I think it's going to be difficult to do that. You might have to put a ring on the microscope slide first. Yeah. And then you have to store the slides horizontally because otherwise the pollen or whatever you have in there will actually sink. Yeah. You mentioned that that the mounting medium contained antibacterial and antifungal components. Could you use UV light to sterilize the slides after mounting? The point is maybe UV light. You see, I think the problem is not the slide itself, but the mounting medium. You see, because every time when you open it up, you actually have a risk of contamination, and then sooner or later you're going to have fungus growing in it, right? So that is actually the real problem. You could use maybe you could use it ultraviolet light, but then again, you'd still have to seal it off, and the problem with this here would not be solved. You'd have to, And as I mentioned already before, some people have made their own glycerin gelatin because they did not want to play around with commercial antiseptics and disinfectants. Used also readily available substances like Listerine, mouthwash, or things like that. Yeah? Maybe even adding a little bit of alcohol would do the trick. Yeah? I can imagine. Yeah. Will you use oil immersion objective since you dry the slide? Will you use oil immersion objective? You dry the slide. Let's put it this way. What I could do, and I'm not going to do that now because I'm running out of immersion oil. And what you can do is you can actually dry the slide and you can heat fix the pollen. Heat fixing the pollen means that you pull the slide through a flame so that the pollen becomes baked and sticks to the glass surface like an egg that you're frying on a frying pan. And then essentially when you add the immersion oil, it's not going to be washed away. So you could actually use oil immersion without cover glass on pollen, provided that you're heat fixing them. But that's something I've not done today. Yeah, you could call it a tardigrade gravity trap. Instead of dissolving with distilled water, you can also use alcohol. Yes, I also, yeah. Basically, I've also read that you can use alcohol, but anytime when you use alcohol, you have to be aware, of course, that the pollen shrinks. So I guess you have to use a proper alcohol water mixture, I would guess. Yeah. It would be nice to have more observation microscopy live streams. Okay, I will do that. Okay, so a little bit, but then again, it's a little bit risky sometimes because you don't know what you're going to see. Yeah? Whenever you use paper towels on the slide, it always leaves little fibers. How can I fix that? That's correct. That's unfortunate. Therefore, what I usually do is I dry wipe it. This one is pretty dirty and old already, but it's a microfiber cloth, and usually this works quite well to remove those cellulose fibers. Yeah. The study of pollen is also used to reconstruct ancient environments. I already talked about that. Yeah. So that's a little bit the importance of pollen microscopy. The whole books have been written about that. Yeah, archaeologists use pollen from excavations. Yeah, I love those videos. Thank you. I suppose you're referring to the new format. It's not that new, but it's kind of an unscripted format. Yeah, where you simply put stuff under the microscope and simply show it to you. A little bit like a live stream. Yeah. Have you ever tried to mount pollen with PVA? Okay, I've talked about that. I already like this stream because it shows me how to do stuff. Yeah, different people have different expectations. Some people like more the live streaming things. Some people like more not live streaming, the observation thing. Some people like more the practical things. I try to use this format deliberately. Also, if going through questions is not as I would say not as observational, I still want to use this format because it's one of the few possibilities how I can directly interact with the community, right? In a live, I mean, that's the whole point of a live stream, that I can somehow interact with you guys. Yeah. Do we need to do similar procedure if we collect pollen directly from a flower? Yes. And why are you not doing the same? Well, I have done the same and I will do the same, but I just ordered the glycerin gelatin and I needed pollen, and basically that's the reason. And that's the reason why I used this pollen over here. But in a few months, when the weather becomes warmer, I'll be going out and I'll be collecting some fresh pollen. And yes, I want to grow some pollen tubes live under the microscope. Still working on that. Yeah. New mini videos are good. Thank you. I'm having trouble looking at the microorganisms on a drop of rainwater. Can you give me some tips? Easy tip: do not use rainwater. Just freshly collected rainwater will not contain a lot of microorganisms. You have to use some standing water from a puddle, from a lake, and not just from the pond, from the water surface, but from the bottom of the pond. You have to collect some solid material, right? So for example, just a few days ago, I went out to a pond, collected some algae, put some of the algae, the green stuff, under the microscope. You'll find lots of things. Okay, now. So pure rainwater won't have so many things. Yeah, so that's the thing. So essentially, wrong place to look, unfortunately. Yeah. I'm a retired cook. Okay. That's the leaf gelatin. I also tried that. Why not? Try it out. Yeah. You don't need a dye, but the dye that's used for pollen, for whatever reason, is called fuchsin. Yeah, fuchsin. I don't know how to pronounce this. Yeah, and that's the one that's used for pollen, and that's also the red one that's in here. But you can use other dyes as well. But I would say if you start out, don't keep it simple. You don't need to add any stains. Yeah, because you're going to see the pollen anyway. So that's the reason. Yeah, I guess the pollen has a tube cell. Yeah, so that's basically what happens is when the pollen lands on the flower. then it starts to grow a pollen tube, and in this pollen tube, they are the cells that will fertilize the egg cell of the female part of the flower. Yeah. Have you seen a pollen tube grow yet? Yes, I have. From freshly collected flowers. But I had problems actually inducing the growth, so I bought some borax because apparently borax is needed, and you need to also have the right sugar concentration, and it doesn't work for all flowers equally well. Yeah, where did I buy that coffee warmer? Amazon. Okay, yeah, Amazon. And honestly, the most of them are with a USB. I did not choose the one with a USB because I wanted to have more power. Yeah. And this one, I don't know. I just bought this on Amazon, yeah. So this is they are not quite as common, but this still should be there. I just keep my distilled water. Would never have so of putting hydrogen peroxide. Okay, that seems to be another issue, other question. Okay. Why did the glycerol in the tip become solid? Well, it's the gelatin that became solid because it cooled out. Okay, that's simply because of the cooling. Yeah, it might be possible, but it's messy. You see, I'd have to put the tip on the coffee warmer. The problem with pollen and diatoms is that they get carried around by wind. Okay, so in archaeology, it's not always a good biomarker. Yeah, so it's a risk of contamination and so on. Yeah, I also talked about this. If you add any water-based medium, and if you put a ring around it to prevent evaporation, it's probably going to stay liquid longer. Yeah, I just wonder how much the water is able to evaporate through the nail polish. Yeah, if it's a complete seal, I don't know. Yeah, and yeah, and that's the last page here. I just got my order with Canada balsam. Oh wow, no U-Pearl available. Any tips for except when it dissolves with xylene, the subject has to be completely water-free? I did not work with Canada balsam yet. It is a traditional mounting medium and is known for, if the specimen is properly prepared, it's, I don't know, the slides last indefinitely. Apparently there are slides mounted with Canada balsam that have over 100 years. So yeah, you got to be careful with the xylene because of inhaling. It has to be completely water-free, but otherwise I'm not able to give you a lot of advice here. Yeah, maybe one advice that's a general advice is make sure that the specimen is basically completely surrounded. Maybe you can put the specimen for a couple of days into liquid Canada balsam so that it's completely soaked all the way through. Okay, that's generally something that you should do anyway to make sure that it's really that the mounting medium also goes into all parts of the specimen. Yeah. What's the size of those pollen? Usually, I think 10 micrometers. Yeah, I'd have to measure it out. Yeah, but they're fairly large. Yeah, a sample I collected in water had many things. A few stood out. There was a single cell shooting balls, and it looks like it was feeding on a single cell. Oh, okay. I wouldn't know what that is. Yeah, but usually, if you take a water sample, you're going to find quite a few organisms eating each other up. That's quite nice to observe as well. Does pollen have a nucleus? Yes, and it will actually, or the nucleus will actually divide and form the pollen tube and also the sperm cell. Yeah. So yes, it's got to have a nucleus. It's a eukaryote and it has DNA. Yeah. We're 20x objective, so I expect relatively big, considering maybe 200 micrometers across in one grain. 200 micrometers would be a little too large, I would say. Yeah, because it would be a fifth of a millimeter. Yeah. So hard to say since the camera sensor and screen size. That's correct. One something that yeah, I'd have to measure it out. Yeah. Fossils, can they be seen under a stereoscope? Possibly. I think it would be kind of difficult maybe to identify them. I remember several years ago they found some interesting structure on a sample of Mars rock. And they were thinking, is this a fossilized bacterium or not? Yeah, and they looked at it under a stereo microscope. Yeah? Can we simply use real raw tree sap as a mounting medium? I would I mean, Canada balsam and Upearl are I think, from plants, but I would say probably not. And the reason is because products that are directly taken from nature often are not very pure, and most likely there are contaminants in there, maybe cells, whatever other things, dust, and so on. So that would probably be the reason, yeah. And usually those mounting media that you can buy, even the natural ones, are actually quite highly purified. Yeah. Do you have the capability for oblique lighting? No, not on my microscope that I have. I've got others where I'm able to do that. But my microscope has certain filters installed that do not allow me to do oblique lighting. Yeah. Is pollen an organism or a cell? Technically, it has cells, but I would not consider it as alive. I would consider it as alive because it is able to form a pollen grain and it is able to divide. Okay, so and it has DNA, right? And it has a metabolism. At least the metabolism becomes active when you add water, and also when it starts to pollinate the target flower, then it starts to become active. Yeah, so yeah, it's a little bit a question of definition, as you can see. Yeah, sciencey fidget spinner. Yes, that's the slide ringing table. Would it help paint a thin ring? Yes, I did that. And sealing with nail polish will extend the quality. I also talked about that. Okay. So what I'll quickly do now is I'll quickly go over again to the comment section, and then I'll simply put again some of the slides again under the microscope. Simply that we're closing off a little bit with some stuff under the microscope. How do I observe bioluminescence? Ooh, gee. That's a challenge. Okay. Under the microscope, you mean? Never tried that. You need, I would say, dinoflagellates that actually do biofluorescence. The bioluminescence, excuse me. Yeah. Do any of your antique slides use this medium? That's a question. Do some of my antique slides did they use glycerin gelatin? And the answer is I don't know, because unfortunately they're not labeled. However, I probably would guess. Let's turn on I probably would guess that most of the antique slides probably would not have used glycerin gelatin, because I'm just guessing, glycerin gelatin is not that super stable, right? It remains semi-solid. I don't know if it's able to last 100 years. Canada balsam is able to last 100 years, right? And many of my slides that I have do not have specimens that actually are usually use glycerol gelatin for organisms that are quite sensitive to dehydration, and I do not have so many of those slides. Yeah, so. Yeah. Did you get my email with my link to my sand images? Yes, thank you very much. I think I got that link. I've got holidays now, so what I'm doing right now is I'm trying to go through all of my emails and trying to answer them. I do have a little bit of my email folders did pile up a little bit, but they did not end up in, thank you, they did not end up in the spam, okay? Could you make a ring with UV adhesive which instantly hardens with UV light? Yes, why not? I never tried it. But generally, that's one of the nice things about a hobby like this is we can experiment. Yeah. And I know that people have experimented with quite a lot, nail polish as a mounting medium, and so on and so on. I would say probably there's nothing that has not already been tried out. Yeah. Would it be useful to use silicone as a mounting media? I don't know. Seriously, if it is transparent enough, if it has the right refractive index, if it's stable enough, I would probably say maybe some people even have used it before. I've not used it. Yeah. So this is therefore something that I would also say is please try it out and share your experiences. Yeah. Any recommendations for sources fluorescent dyes? Normally, if you want to use fluorescence, you're using antibodies, expensive. But what you can try is, and I've done this as well a long time ago just for fun, there are those fluorescent markers, not the blue ones. but yellow ones, and actually they will shine. And I think sometimes you are able to use those. I read it as well somewhere to actually do some fluorescent staining, the yellow markers. Yeah, not tried it out. I once experimentally tried this out, and it did start to shine a little bit with some UV light. But it's not something that I've done where I could actually say that I can recommend it, or that where I can give you more information about that. Yeah. We should try mixing PVA glue with glycerin. I tried that actually to get different long-term properties. So Alexandra is asking this. I actually tried that to mix PVA glue with glycerin, and it did not harden fully. It kind of made a very smeary, slimy consistency. So maybe I didn't get the proportions right. But this actually also has been done already, mixing PVA mounting medium or PVA glue with glycerin. Yeah, so to adjust primarily the refractive index. Yeah. So another great presentation. Thank you very much for the nice feedback. What does your jelly taste, smell like? Would take a bite out of curiosity for sure. I would not. This glycerin jelly here, right? Because I don't know exactly what substances they added as a disinfectant. That's why I wouldn't take a bite out of that. Yeah. So let me quickly go through here. Yeah, the cookies might have some preservatives that keeps the slime mold away. It could be in this specific case with the slime mold. I don't think that there were preservatives present because the students made those cookies and they sold them for Valentine's Day. So, yeah. Okay. I hope I did not forget anything here. Okay. There's so many comments over here. If I skipped over some of your comments, then please apologies. Okay, but I think I'm slowly going to call it a day again. Yeah, here these are now the. Just a second. These are now the. Let me see. Yeah, these are the freshly mounted pollen grains. So not and you can see that they are now turning red. Yeah, so these are the ones that we just made before. Yeah, so. Yeah, here let's go up a little bit. Let's try 60 times just for the fun of it. And you can see that they basically look quite nice, bright field now. Yeah, and here this ring here at the top. Do you see this? Yeah, there's I can adjust for the thickness of the cover glass to squeeze out the last bit of resolution as well. Yeah. Okay. Yeah, I think I'm going to call it a day now just a 2-minute summary for those of you who joined in late so that you know what I've been doing. I bought myself some glycerin gelatin over here for mounting pollen. I warmed it up using a coffee warmer over here, right, at 40 degrees. Do not overheat, not hotter than 40 degrees. And I made a couple of pollen slides. I got the pollen from the pollen basket from some of the bees that were sent to me from Microscopy friend in Germany. Thank you again. Yeah, and basically this was a little bit of a more challenging type of preparation because I also tried to do a couple of these slide rings here. Not very nice, but at least you get the idea. Yeah, and. Basically, I just wanted to try this as well, not only because it was a request by one of my viewers, but because also it's a slightly different way of preparing microscope slides. And basically, that's the result that we have here. Yeah, these are then the pollen, which now start to slowly turn red because of the pigment inside the mounting medium. So maybe that's also a little bit of a new concept that there are now that you can buy mounting media that already have a stain mixed in right from the beginning. Enough for today. And yeah, can you see the nucleus of the pollen? Last question. I don't know. Probably. I don't know if this is nucleus or not. Maybe it is possible to make it visible to use some kind of a stain that stains the DNA, right? or maybe one of these things is the nucleus. I don't know right now. I could not be able to identify it. But in any case, what I'm going to do now is I'm going to call it a day. Yeah, happy microbe hunting as always. I'll think of another topic for next week, maybe as requested. I'm going to do again some observation of some water samples. And I wish you all the best. Happy microbe hunting as always, and bye bye. And have a nice weekend.